Today in this class I am explain about patenting of gene and DNA sequencing and patenting of transgenic organism. A patent is an exclusive right granted by country to the owner of an invention to make, use, manufacture and market the invention. This right implies that no one else can make, use, manufacture, and market that invention without the consent of the patent holder. And patent represents a contract between an inventor and society. And patents are a form of intellectual property. And what is intellectual property? Intellectual property refers to a creations of the mind, and intellectual property is divided in two categories. Industrial property includes patents of invention, trademark, industrial designs and geographical indications, and copyrights. And copyrights covers literary works such as novels, poems and plays, and films, music, artistic works. For example, drawing, painting, photographs and sculpture. And intellectual property is a legal term, and intellectual property rights are the rights given to persons over the creations of their minds. They usually give the creator an exclusive right over the use of his or her creations for certain period of time. And next topic is granting patents. A granted patent application must include one or more claims that defines the invention. And a patent may include many claims, each of which defines a specific property right. And these claims must meet relevant patentability requirements such as novelty, usefulness, and non obviousness. And this exclusive right granted to a patentee in most countries is the right to prevent other, or at least to try to prevent others, from commercially making and using, selling, importing or distributing a patented invention without permission. And what can be patented? Today, patents have been granted for a range of different biotechnological processes and products. It includes therapeutic, therapeutically active proteins and monoclonal antibodies and microorganisms, transgenic organism, and genetically altered microorganisms such as bacteria, fungi, algae, virus, and enzymes, and nucleotide sequences derived or isolated from the human body. And next topic is gene and DNA sequence patenting. Genes are defined as a region of DNA. A gene is a unit of heredity that is transferred from a parent to offspring and is held to determine some characteristic of the offsprings. And human beings share mostly the same genes. In the case of gene patents, firstly, genes are naturally occurring. We cannot patent snow eagles or gravity and we should not be able to patent gene. And while much intellectual effort may have gone into discovering them within the DNA sequencing, by now 1/5 of the genes in our body are privately owned. If the genes are isolated or otherwise produced by means of technical process, they can be patented. And the human DNA and nucleotide sequences may be patented when they satisfy the basic criteria of patentability. They have to be novel, inventive, and industrially applicable. The first one, novelty. A gene in a body cannot be patented, and the patentability of human genes is thus limited to artificial structure outside the human body containing the information about the human genome. And the inventive step. And inventive steps. Inventive step means a feature of an invention that involves technical advancement as compared to the existing knowledge or having economic significance or both, and that makes the invention not obvious to a person skilled in the art. And next one is industrial applicability. For example, therapeutic use. The earliest genetic patent was issued in 1982 following the US Supreme Court case of Diamond versus Chakrabarti, which opened the door to patenting biotechnology discoveries, and submitted an application to the US Patent Office in 1972 for a new strain of the bacterium Pseudomonas. The novel bacterium were intended to clean up oil spills in water by degrading the crude oil. For example, Canava disease is an inherited disorder that affects children starting at 3 months. They cannot travel or walk, became paralyzed, and die by adolescence. And formerly, there was no test to tell parents if they were at risk. Families enduring the heartbreak of caring for these children engaged a researcher to identify the gene and produce a test. Canava families around the world donated tissues. When the gene's mutation in ASPA gene caused Canava disease was identified in 1993, the families got the commitment of a New York hospital to offer a free test to anyone who wanted it. But the researcher employer Miami Children's Hospital Researcher Institute patented the gene. The parents did not believe genes should be patented and so did not put their names on the patent. And next one is patenting of transgenic organism. The organism whose genetic makeup is manipulated using recombinant DNA technique is called genetically modified organism or transgenic organism. In several countries includes USA, Japan, Europe, etc. The transgenic organisms can be protected through patent, and animals and plants have been modified through breeding programs for many centuries, and transgenic animals or plants can now breed with specific genetic characteristic. This helps in the following way. First one, to investigate new medicine. Second one, can produce therapeutic proteins. And third one, other techniques of genetic engineering of an animal make possible test of promising new cancer treatment. And fourth one, improve the food supply as well as quality of plants. And fifth one, disease or insect resistance plant. Both transgenic animals and plants are patentable for this production characteristic, and patenting of transgenic animals and plants is justified to prevent copying, and it is not possible to patent life. And the patenting of transgenic animals has been safeguarded from the of ethical point of view also. And before any animals experimentation can be undertaken, license are required. For example, genetic engineer Ananda Mohan Chakrabarti, working for General Electric, had developed a bacterium and derived from the Pseudomonas genus and now known as Pseudomonas butida, and capable of breaking down crude oil, which he proposed to use in treating oil spills. And the General Electric filed a patent application for the bacterium in the United States and listing Chakrabarti as the inventor. And the next one is the Anko mouse or Harvard mouse is a type of laboratory mouse that has been genetically modified using modifications designed by Philip Leader and Timothy A. Stewart and Harvard University to carry a specific gene called an activated oncogene. And the activated oncogene significantly increases the mouse's susceptibility to cancer and thus makes the mouse suitable for cancer research. And glowfish, the first genetically modified and the animal to be sold as a pet. And the next topic is ethics. The patenting of genes is a controversial issue in terms of bioethics. First, some believe it is unethical to patent genetic material because it treats life as commodity. Second, some say that living materials occurs naturally and therefore cannot be patented, while some feel that the patent on living material is unethical. Others believe that not allowing patents on biotechnological inventions would also be unethical. Another area of controversy in genetic patenting is how gene samples are obtained. The question of benefit sharing also arises when obtaining genetic samples, specifically the potential responsibility of the collector to share any benefits or profits of the discoveries with the population or person from whom the sample came. And since the evolution of patent legislation, there has been a greater input on the role of ethics in the patenting process. And the patents have been granted by India for different product. A process for the preparation of an herbal therapeutic product extracted from the pulp of the species Eugenia jambolina in 2002, and a process of isolation of oligosaccharides having immunostimulating activity from donkey's milk in 1998, and a process for the preparation of a composition used for the detection of latent fingerprints in 1996, and a herbal anti-diabetic drug in 1999, and genetically engineered clone of hepatitis E virus genome which is infectious, its production and uses in 2000, and a process for the preparation of an extract of Annona squamosa of the treatment of diabetes in 2003, and last one is earthquake alarm in 2001. That's about the patenting of gene, DNA sequencing and transgenic organisms. Thank you.